Okay, so thank you again for joining. My name is Eileen Fitzgerald, she, her, and I'm the Education and Outreach Associate with the Lake Champlain Committee. I'm joined today by Meg Modley of the Lake Champlain Basin Program, as well as by Dr. Lindsey Carlson, the LCC Water and Science Coordinator. Um, just to go over some Zoom things quick. I already mentioned most of this, but this presentation will be recorded and shared with volunteers, so please remain muted with video off until the end of the training. You can mute your microphone with those icons there. Um, and as you go, if you are thinking of questions, you can pop them into the chat function, and I can either answer them if I see them throughout the presentation, same with Meg, or we will definitely get to them at the end during the Q&A. So this training should be about an hour and a half. We're going to be covering a lot of ground in the training. So just something to remember up top is that you don't need to memorize anything that we're covering right now, since everything will be shared in the champ manual and it'll be reiterated for the in-person trainings, and a recording of this training will be available to you. And not only that, but I will be available to you all season long with any questions that you may have. But what we're covering in this training are just an overview of what aquatic invasive species are. You'll see that acronym AIS pop up a lot. That's what that means. Why they're an issue in Lake Champlain, what we can do to prevent the introduction and spread of AIS, and what this program is as a volunteer monitoring program to look for aquatic invasive species in Lake Champlain, and what those species that we're looking for are specifically. That's where Meg's expertise comes in. Um, and then I'll go through step by step how you do a survey for aquatic invasive species with CHAMP and how you send that survey to us. So I'll pass it off to Meg for these next few slides. Thank you.
Great. Thanks, Eileen. Can you hear me okay? Awesome. Thanks. Okay. Good afternoon, everyone. Thanks for joining me in my water chestnut pond here. This is actually the southern end of Lake Champlain. And those of you who have been down there may be familiar with the invasive water chestnut plant. But to talk about invasive species, we are using a federal definition which defines aquatic invasive species as plants, animals, or pathogens that are non-native to the area and are documented to cause some kind of harm. And that harm can be to the economy, to the environment, or to human health. These invasive species usually are introduced into spaces that have no competitors or pathogens from their original or native place of origin. So they're able to reproduce really prolifically. And some of the top invasive species present in Lake Champlain include the Eurasian water milfoil, which is the left-hand photograph. That's a whole, nearly a mono stand of Eurasian water milfoil growing up to just a few inches below the water's surface. Um, the invasive fishhook and spiny water flea that are present in Lake Champlain, and those are thousands of them glommed onto a downrigger or a fishing line that's been pulled through the water. And just to clarify, they don't seek out the line, they just get hooked on it. And then when the anglers pull up their lines, they become gunked or fouled, and we have to remove them. And then many of you are probably familiar with the zebra mussel, which has many impacts to the lake. It filters a lot of biota out of the lake, so it makes the water look clearer, but that does not mean that the water quality is improved. But these invasive water chestnut, zebra mussels can cut the feet of swimmers, clog water intake pipes, and foul our historic shipwrecks, as well as grow on top of and suffocate native mussels. So there's a number of different impacts from these three primary invasives from Lake Champlain. Next slide, thanks. And then ways by which these species move across the landscape. These invasives usually come into our systems with some human aid. We have built canals that connect watersheds together. So Lake Champlain is now connected to the Hudson drainage with the Champlain Canal that crosses the watershed divide. And without it, plants, animals, and pathogens couldn't swim or be entrained or move across that watershed without that pathway being provided. There are bait fish introductions that can occur. Some of these are intentional, some of them are accidental. It's the whole reason why we had the emergency fish regulations go into effect in the late 2000s that require disease-free certified bait and that we don't move them between water bodies or we have corridors approved for where they can move. So one of those species depicted here is the invasive alewife that got into Lake Champlain and is causing a number of issues for our native salmonids, and they're very susceptible to die off. So we do sometimes get some stinky shoreline accumulations of alewife along Lake Champlain. Water guarding is also a common, often unintentional way that these riparian or aquatic invasive species can become introduced to a water body. We may be familiar with the flooding of the last few years, or like Tropical Storm Irene and Lee that came up. And with these instances, we have species planted in back gardens that quickly became flushed into side ditches and came into local lakes or into streams and have fouled the waterways. One of the biggest species that spread during some of these flooding events have been Japanese knotweed. And we've seen a lot of uptick of that species in both Vermont and New York across the watershed. Overland transport on boats and trailers. This is often, again, unintentional. Aquatic hitchhikers can attach to or live in very small amounts of water that could be trapped in live wells, could be on anchor lines, could be in bilges. And oftentimes when boats come and go from the lakes, there's aquatic vegetation growing near the launches, and those plants can become impinged between the boat and the trailer. So we have the Watercraft Inspection and Decontamination Program. There are stewards all across the Adirondack Park. They flank Lake Champlain on the New York, Quebec, and Vermont side, and we have the greeter programs in the state of Vermont to address overland transport of invasive species on boats and trailers. And finally, aquarium dumping is another popular one. I call it the free Nemo phenomenon, but oftentimes when pet owners have these pets that either get too big for their spaces, their cages, their tanks, or they're no longer able to take care of them. They have a very well-intentioned thought of returning them to the wild to live out their days. However, sometimes this can lead to introduced reproducing populations of invasive species. And sometimes we see goldfish reproducing and living in the retention ponds around development centers like at Taft's Corners in Williston, Vermont, and that's been a situation that's had to be dealt with. Next slide. So we try and paint a picture of what we know about what's present in Lake Champlain and how we are connected to the water bodies that surround us. Currently in Lake Champlain, we have 51 known non-native and invasive species that are established in the water body. And the difference here is the darker green colored species are known to cause some kind of harm. So we can pull out purple loosestrife, water chestnut, Eurasian water milfoils, zebra mussels, spiny and fishhook water flea. Those are species that are not native and have been measured to cause some kind of harm. Then we have a whole slew of non-native species that are either close to the tipping point of being able to be measured to cause some kind of harm, or were intentionally introduced for our enjoyment, like brown trout and rainbow trout, for example. Non-native, not from here, we put them in. They're not part of what was here before we changed it. So that's what we know from looking at what's going on in Lake Champlain of what is non-native and might be causing some kind of harm. And then adjacent to that, we have documented the number of known non-native and invasive species that are present in surrounding waters and watersheds. So the Great Lakes are home to over 190 species, the St. Lawrence 95, the Hudson 146. Those overlap primarily with the 51 that we know occur in Lake Champlain. It's important that we do experience some degree of buffering or isolation from these larger numbers of aquatic invasive species, largely because we are connected to those systems by canals, and it takes extra effort for these species that have now been introduced into these systems to hitchhike on a boat and trailer, be transported in a bait bucket, or swim through canals, for example, to make their way into Lake Champlain. But what once present in Lake Champlain, now we're the source for overland transport or other pathways of introduction to uninvaded water bodies throughout the region. So Lake Champlain is viewed as a source lake when we think about just the watershed of Lake Champlain. So why are these species an issue? I think I began to touch upon it, but the rate at which these species are introduced and the fact that they don't have, again, the predators that exist in the habitats and that they came from means that they have a competitive advantage. And most introduction of invasive species or non-native species will fail, but those that are really great ecosystem engineers, like water chestnut in my background, or the zebra mussel, or the alewife, these are species that are able to tolerate a range of different uh, parameters of water. So, like temperatures, dissolved oxygen, pH. There's a wider range that a number of these aggressive invasive species can tolerate. So, they can reproduce early, often. They can take advantage of light. If you're a plant, they'll come up first. So, the first invasive species we see in Lake Champlain and at this time is Eurasian water milfoil and curly leaf pondweed. We're seeing a lot of ahead of our native species that are still waking up from the winter. So they are also able to live in a number of different habitats. So whether they're sandy or they're hard substrates, these species are just able to adapt and take hold a bit faster and outcompete our native species. And so at what we hear and what we experience in these water bodies, if we have these very dense beds of water chestnut, it's really difficult to boat, swim, or fish in that bed of aquatic vegetation. There's also not a lot of native plants growing there, and the dissolved oxygen under that bed of water chestnut is near zero. So it's also not providing habitat for other species. So as they monopolize and they crowd out, um, it really limits the amount of habitat, spawning habitat, or food availability for our native species. In addition, some of these plants and animals can carry other diseases or toxins that are then very harmful to the native species that we have in our ecosystem. So it is a shock, and the rate of introduction and the tolerance or the range of habitats that these species can occupy makes them such great invasive species. So what can we do? There's a lot that we can do, and we try to work with other regional partners and nationally across the United States in using different kinds of education and outreach messaging. There is the Don't Let It Loose campaign that addresses releasing of pets in aquarium species into the wild. It's about returning the species or even considering euthanizing some of them to protect the native species and habitat of your wherever you're living. There's a clean, drain, dry messaging about removing, cleaning off, and properly disposing of any water, plants, mud, or other materials that could be hitchhiking on your recreational watercraft, your trailer, or other equipment. And there's an accompanying Stop the Aquatic Hitchhikers campaign. So these are different messages targeted at different users in the watershed. There's Don't Move Firewood, there's No Before You Go. There's quite a number. There's Play Clean Go. There's a lot of different national messaging that we are subscribing to, especially in this watershed, to really promote best management practices when we're out and utilizing our waters.
Okay, so knowing all that about aquatic invasive species, we're now going to talk about what this program is and how we're addressing that problem of aquatic invasive species. So CHAMP is a community science program run by the Lake Champlain Committee. LCC, that's Lake Champlain Committee, is a watershed-based nonprofit dedicated to using science-based advocacy, education, and collaborative action to safeguard habitat, promote access, restore water quality, and foster stewardship in the Lake Champlain watershed. A major component of our work is community science, which is a type of research that provides everybody who signs up, regardless of background, an opportunity to contribute meaningful data. LCC manages the cyanobacteria monitoring program as well as CHAMP. Some of you here today are maybe volunteers with that program as well. But community science allows you, people who are participating, to be the eyes and ears on the ground for us, as well as for those who manage aquatic invasive species populations and for researchers trying to better understand their introduction and spread. CHAMP, we started CHAMP in 2023 in response to state agency need to survey for aquatic invasive species on Lake Champlain. Vermont and the Adirondacks both have really solid volunteer monitoring programs for aquatic invasives, but they're just since the lake is positioned right between those two regions, there are some issues getting coverage on the lake. And that's where we stepped in with support of the Lake Champlain Basin Program as well as NUIPIC, and we piloted the program in 2023 and have since grown it. It's now 2026, and we're going to be covering dozens of sites throughout Lake Champlain, all thanks to volunteer effort. So thanks. And through CHAMP, we train volunteers in aquatic invasive species identification and survey methods, and we give them the tools to conduct surveys and report their findings to us. And then these findings inform research, management, and very importantly, early detection and rapid response actions. So what I mean by that is when there's a new aquatic invasive species that is discovered through something like CHAMP, there's this set of protocols that are followed to follow up on it quickly before it becomes an established population. And we saw that in action. In 2024, we had a CHAMP volunteer who was the first one to find golden clam in Lake Champlain, which just sort of reinforces how great it is to have this many eyes and ears on the lake throughout the season. And you'll be learning in this training, we have 8 target aquatic invasive species that we know are in the lake and 8 watch list aquatic invasive species that have not been detected in Lake Champlain, but they pose a threat of invasion. I'll talk a little bit more about that shortly. And you'll also learn step by step how to conduct and submit CHAMP surveys. So as a CHAMP volunteer, you'll participate in or watch this virtual training session. So those of you who are here now are like maybe a quarter or a third of the way done with that. And you'll also attend an in-person training session in mid-June. This is something you should have registered for on your sign-up form. If you're still not sure about when that is, please let me know. And then once you have both of those trainings done, you are considered good to go to begin surveying. So it'll be like late June when you'll be able to get going. And we ask that you conduct and submit 3 surveys for aquatic invasive species at your site throughout the season. This comes out to about once per month. If you skip a month, we accept surveys all the way through October. So there's some wiggle room there. And LCC supports you with these trainings, as well as a toolkit, which is all the physical materials you need to survey. That includes the manual, which has a chock full of identification resources on all of the species you're looking for in CHAMP, instructions on how to do a survey front to back, guidance on how to submit a report, and contact information for myself and anybody else you need to contact for CHAMP. We give you these aquatic species rakes, which are tools that help get aquatic biomass to you. I'll talk more about that later as well. We give you a container that helps you identify the species against a white background and photograph them. You also get a tote bag, so that's nice. And as I mentioned earlier, ongoing technical support throughout the season. So you can always contact me with a picture of a plant or any questions about a site or anything like that. So, what are we surveying for? Um, I alluded to this earlier. We split the species that we train you in into two groups. There's target species, which we know are in Lake Champlain to some extent. Some of these are very well established in the lake. Some of them, there's only a couple, not even really a reproducing population. I'm talking about golden clam. Um, but the other group is the watch list species. So these are species that are not detected in Lake Champlain, but as we saw in that map that Meg was talking about earlier, there's many other species sort of at the doorstep when we're looking at water bodies such as the Hudson River or the Great Lakes. So these are species that are established there or in other adjacent water bodies that we want to make sure that we catch early should they be introduced to Lake Champlain. So looking at the target species, these are the eight that we are looking for. And you may be familiar with these already. Even if you're a new volunteer, you may be familiar with a few of them. And these are the eight species that you would be more likely to encounter in your CHAMP surveys, just because we know that they are to some extent in Lake Champlain. And I will pass it to Meg to do sort of a breakdown species by species of how you can tell what it is when you're in the field.
Great. Thanks, Eileen. So we're going to start off with brittle naiad, and brittle naiad is a species that grows really close to the bottom of the lake. It doesn't grow far off the bottom. Brittle naiad it's really looks like a feather poof, but it has serrated lance-like leaves that don't dissect. They don't split apart. So this plant we don't see growing in long strings. We see them growing really close to the bottom of the lake, so near the sediment. And so if you have a population of brittle naiad, it's likely growing near the shore when you're throwing the rake, but it's going to come up in the mud more than you would, a Eurasian water milfoil strand might be all the way to the surface. So this one, they're shorter, they're spiked. So let's go to the next slide. And we do have some native naiad species, but this one is really told apart by the serrations. And that's going to be a common theme throughout all of these invasive species that we're looking for. These spikes and serrations on the leaves are more common for the invasives here. So thank you for flipping ahead. We have you can see that the leaves come off opposite each other, off the stem instead of alternate. So opposite, and they sort of curl out and they have these spikes or serrations again on the leaves. So if we can just go to the previous slide before the last brittle naiad. Let's go back just one more. Just give you a little bit more about. So most of these are coming from Eurasia. This one is coming from the Southeast US and South America. It's a little different. So it's been confirmed in only a few water bodies in the watershed. It is in Lake Champlain. We do see it in Moncton Pond, and it's confirmed in other states around New England. So it is possible that you will see this species depending on where you are in Lake Champlain. I wouldn't say it's widespread throughout the lake, but you will probably find some this field season. And then we'll skip over to curly-leaf pondweed. Oh, these are native lookalikes. Great. Sorry, missed this one. So our native naiads or slender naiads are similar in appearance. So again, the leaves are opposite, but they do not have that spike on the leaf. So the brittle naiad is also, it feels a little bit more firm, like it has more structure to it. And if you run into native naiad, they're much more delicate. So again, if you come upon something that looks like this, and you can key it out to be a naiad because it has opposite leaves that are lance-shaped. The brittle will be spiked, the native will not, and it will be growing again closer to the sediment interface. And then you can always look at it with a hand lens or give Eileen or myself a photograph or a sample to help confirm what you're looking at. Great. So curly leaf pondweed is the only invasive pondweed that we have in Lake Champlain. We have a lot of native pondweeds. So Potamogeton crispus is really well known for its lasagna noodle-like appearance in the water. So these leaves you can see some can have a very curly or wavy edge. Our boat launch stewards are already intercepting curly leaf pondweed, and the leaves that are lower down on the stalk tend to be not as wavy as the ones closer to the surface. But this is a species that was introduced from Europe. We do have it throughout Lake Champlain. I would be surprised if you don't find it wherever you are around the lake this summer. So it grows in really dense beds in certain areas of the lake. It's just a monostand and there's nothing else there. But it is widespread across New England. And I think once you start to see this one, or if you saw it last year, it should be pretty familiar when you see it again. It also has these buds at the tip that are bract-like, and it looks like sort of an upside down flower or flower petal, but it's really hard and firm. And so that's another telltale piece that you've got a piece of Potamogeton crispus. But when you look closely at the leaf, again, you're going to see those serrations along the leaf edge. And that's a good indication that you're dealing with an invasive pondweed or curly leaf pondweed. Okay. Feel like we're going fast. Eurasian water milfoil or Myriophyllum spicatum is very common in Lake Champlain and in inland water bodies. We do have a challenge with our milfoils, and I'll get to that issue, but it's because of hybridization. So some of the milfoil that you encounter will look very much like it does in these photographs, where it's long and stringy. And it has a very long internodal space. So that's the space between the whorls that come off the stem are elongated compared to native milfoil species. This one is again from Eurasia. You can see it's present in many different water bodies across the watershed and across New England and into Canada. Um, the hybridization is tricky because the spicatum can hybridize with our natives, but we also have a variable leaf milfoil present in Lake Champlain. So here you can see these feather-looking leaves. There's 4 leaves on a whorl, so there's 4 of those feathers that come off at one point around the stem. And the telltale for the Eurasian or an invasive milfoil is that there's greater than 12 little leaflets that come off the side of the feather. If they're fewer than that, it's much more likely to be a native or an early development stage of a variable leaf milfoil. And these feather-like leaves, often people report that they look as though somebody's taken scissors and snipped the end of the feather off, so it's sort of flat. That's not true all the time, but when you do see this present, it can be, when you add it up with four feathers or 4 leaves on the whorl, greater than 12 leaflets on one side of the feather, a greater internodal space between where these whorls come off the stem, you can make it add up to be pretty clearly Eurasian water milfoil. All right. So going ahead to European frogbit, Hydrocharis morsus runni. This one is present mostly in backwaters, sort of the wetland areas, the slower moving waters around Lake Champlain. So some of you will definitely see this. We are fighting an infestation in Town Farm Bay and Charlotte in Vermont, particularly the Lewis Creek Association has been doing a lot of harvesting of this species. It's a free floating plant that is not rooted to the bottom. So that's one of the ways we can tell it apart. It also has these really continuous heart-shaped petals or leaves there. So again, this one's from Europe. It's in Lake Champlain. It's in a number of inland water bodies. It's pretty widespread. It's pretty difficult to control. Let's go to the next slide. So here you can see in the black and white photograph how they reproduce by sending stolons or stems out from one individual plant. And these plants come up from seeds that overwinter in the sediment. So then they will sprout and come up to the surface and then they'll start growing, but they're not again rooted to the sediment at the bottom. They have this nice, pretty white flower with three petals and a little yellow in the middle. That's a good way to tell them apart from some of our native lilies that are rooted to the bottom and usually connected to a big rooty rhizome or their root mass. And then they're all interconnected here too. And that's the other way you can tell this one apart from any of our native species. The leaves themselves tend not to get much bigger than the size of a quarter. Okay, the golden clam or Corbicula fluminea, as Eileen reviewed, was first found by a Champ volunteer down in South Bay on Lake Champlain. This is very different from our native clams in the lake because that first photograph shows really beautifully these raised concentric ridges. So if you could imagine taking your fingernail and rubbing it over the shell, you would hear like clicking. And our native clam species do not have these raised concentric ridges. So that's kind of a telltale for for the golden clam. This species is a hermaphrodite, which means it only takes one of them to reproduce. They can produce many times during the year and can get to great densities. We've been tracking populations in Lake George and Lake Bomoseen that have spread but are not becoming so dominant in the ecosystem that it's easy to measure their impacts. But it is really hard to get rid of these once they show up. And we know they're making some changes to the ecosystem. We're just having a hard time measuring it. The shell colors can change in some bodies of water depending on what kinds of nutrients are present. The shells can be purpley. They can be yellow. They can have some alternating look, not as much as zebra mussels, but there is some variation in their shells and their coloring. And then again, they don't get much bigger. We've seen some as big as a quarter, but usually they're smaller, kind of the size of a penny. So they tend to prefer sandy substrate. Ironically, where they were found in South Bay, I think that's a figment of them being closer to other sources of populations of this golden clam. It's more mucky. There's a lot of sediment and aquatic vegetation growth in the water and detritus in the south end of the lake that acts more like a river than a lake itself. So in places where there's really clear water and there's nice sand, this species can grow in incredible densities, but we have not yet seen that in definitely not in Lake Champlain. We haven't found them anywhere beyond the South Lake boat launch, but we know they're present in the Champlain Canal. They've been fighting the infestation in Lake George, and we know they're also in Lake Bomoseen as of 2016. Okay, variable leaf milfoil. This one's tricky. And it definitely, I think you're going to be able to determine that this one is not a native milfoil again because of similarities to the Eurasian water milfoil. But unless you have a really long strand of this species, it's going to be hard to decipher it between variable and Eurasian. So what you're seeing here in that first photograph is a picture of the bract, which comes up out of the water on top of these sort of variable leaf looks much more like bottle brushes in the water than Eurasian water milfoil. So, but that bract is very rigid and it is brittle and it's a bit unique. Eurasian water milfoil also has bracts, but the variable leaf is pretty prominent. Let's look at the next slide. So variable leaf milfoil, that top left photo I think is a really good one. When you get these dense populations of variable leaf, they really do look like these beautiful laboratory bottle brushes. And they have, unsurprisingly, a varied number of leaflet pairs and leaves around the whorl on the variable leaf milfoil. It was first found in Lake Champlain in 2011. We had a population that we found in the northern end of the lake near Missisquoi Bay, and another population in the southern end of the lake near Ticonderoga. Now, this plant has been present in Lake Champlain probably for a few years before we found it, and it's been well over a decade since then. So, with the currents and things moving around, it could have moved considerably around in Lake Champlain. So widespread, but not that common in the state of Vermont. And that may have to do with luck. That could do with water quality parameters. We'll see. Um, next next slide. So other submersed lookalikes, when I talk about that bottle brush appearance, on the right side of the screen is our native coontail. And the coontail has a different look. When you take it out of the water and you look at the individual leaves, you can see that they're forked. And that forked leaf shape should give it away that it's not a milfoil, because the milfoil, again, has those feather leaves and usually four on a whorl. The coontail is also, when you take it out of the water, got a lot more structure to it. It's quite a bit more brittle. I would expect you to all see coontail this field season in Lake Champlain, another very common native plant, thank goodness. And the plant on the left is one of my favorites. Utricularia species, or bladderwort species, do have branched leaves, and that's different than a feather. So these individual leaves branch multiple times. They can appear feathery underwater, but when you get them out and you lay them out, you can see them. The other telltale on bladderwort, it has these little pods, and the pods are more light green or translucent when they're empty, and they're darker brown or black when they're full. And it is a feeding mechanism of the plant. It is able to capture certain nutrients and little critters in the water to eat. So the bladderwort is it's a really beautiful and really neat plant, but it definitely, when you take it out of the water, does not look like milfoil. Okay, and water chestnut. So this floating aquatic invasive plant that looks sort of like doilies on the water surface, this one's mixed in with some native duckweed, is a pretty telltale plant again. Look at those really large serrations around these triangular leaves. And this plant floats on the surface. It is rooted to the bottom. We'll look at the next slide. The stem of it can almost look milfoil-esque, but when they grow, this vegetation, the structure is much larger than milfoil can grow to be. So we're holding up a rosette of milfoil, and you can see the individual triangular serrated leaves, and those little leaf petioles have an inflated pocket, and that has air in it that helps it float on the water surface. These have been introduced through the Champlain Canal to the southern end of Lake Champlain in the 1940s. We spend about $800,000 managing water chestnut in Lake Champlain and across the watershed, primarily by hand harvesting and mechanically harvesting the species in the southern end of the lake. It is widespread. It's becoming much more of a problem in the Mohawk and Hudson rivers of late. So there's a lot of effort out there to battle this plant. It's an annual, which is that's okay. It's an annual, which means that the nutlets, or when they're dried, they look like those hard black spiky growths on the underside of the plant. They can stick your fingers. They're kind of nasty. You need to know what you're dealing with when you're harvesting water chestnut. They're green when they are ripe and quite alive, and then when they dry out, they turn black. But that is the seed that drops at the end of every year. And if you can get the plant out of the water before the seed drops, you can really work at starting to reduce the population. The challenge here is that those little nutlets can overwinter in the sediments for up to 8 or 10 years, and a very small percentage of them, but still a viable percentage of them can still sprout after that time. So they do have this little white flower that has four little petals. You won't need the flower to help tell it apart. Just its floating structure alone will show you what it is. But I do like the picture on the left in the white tray with that background. Like Eileen said, they'll be giving you white trays to take photographs of these plants. And this is why, because you can see the structure of that plant so well. I can see all the leaves, I can see the stem, I can see the nutlets, and it's not getting confused with other things in the background there. So that's a really great example of why we use the white trays to take the photographs. And zebra mussels, Dreissena polymorpha. Our dreissenids have been, again, in Lake Champlain since the 1990s. They were introduced, again, we believe, through the Champlain Canal into Lake Champlain and colonized the rest of the water of Lake Champlain in about a decade. They are great fowlers of many things. This is a very encrusted lower unit and entire motor of a boat. So these zebra mussels, when they're small, they're called veligers. They float in the water and they move with currents. But they can also attach with a byssal thread to hard substrates, like motorboats. And so, if you have equipment that sits in the water for any length of time, the zebra mussels will seek it out. What they're looking for is a hard substrate to hook onto so that they can comfortably open and close and feed and filter water. And they'll filter between 1 liter to 2 liters of water a day. And that's why there's a lot of public response that the water has become clearer after zebra mussels were introduced. And that's true, but it's not that the water has become cleaner. And what they're doing, in fact, is bioaccumulating a lot of the food source for our native species at the base of the food web, where it can't be accessed. And that is one of the implications of a zebra mussel infestation. So they are D-shaped, and an individual zebra mussel, if you put it up on its side, should be able to sort of stand on its own. And that's important for differentiating it from the quagga mussel when we get there later. They don't get very big. They have many different patterns and like variations of scale between white and black throughout the lake, and they're dependent on calcium. So a lot of the lakes where maybe zebra mussels might have been introduced but are not able to reproduce and survive is likely due to lower calcium levels. That's a limiting factor for them and their reproduction. So they've been present. They like to attach to things. We really want to prevent their spread to inland water bodies throughout the region. So here's a good graphic about the life cycle of the zebra mussel and how many times and how many they're able to reproduce. It is astonishing. So the females, again, can get going when the water gets above a certain degrees, in this case, 50 degrees Fahrenheit. These juveniles or veligers will float and hatch, and then they can travel distances with the currents and things in the lake. And then they can also they can go unnoticed in bilge water or in the motor water or in a live well and be transported to another body of water without even seeing them. So we call them small-bodied organisms that are not visible to the naked eye, which is why when you encounter them, they will likely be attached to a rock that you pulled up from the bottom. Oftentimes, they're attached to the stem of aquatic plants when you rake them out of the lake. And so, take a close look at those plants in particular when you get them out, and look at the stems because you may have some really young zebra mussels growing along the stems there. And yes, they're really great biofouls. So they are clogging water intake pipes. We have issues with them at the fish hatchery. They are in really fouling our historic shipwrecks on the bottom of Lake Champlain. They live for a few years, they reproduce prolifically, and we really want to prevent them from spreading. And we'll talk a little bit about the quagga mussel in the next section. So moving on to the watch list species. Do you want to review these, Eileen, or you want me to?
I think you got it.
Okay, great. Okay. So watch list species are just that. These are species we are on the lookout for, meaning they are not present in Lake Champlain that we know of. And again, that's a really important thing that we know of. And we can't be everywhere. And that is such an important part of this program. And the need for this program is having trained eyes who go out and actually take the time to look repeatedly throughout the year at certain sites because we just do not have the staff to go out and sample everywhere around Lake Champlain. So this is really valuable information that you're gathering. So these are species that we want to warn you about. They have some native lookalikes, but we want to give you some of the key features of these plants and a few animals that we hope we do not encounter, but if you do, it will trigger, as Eileen said, our rapid response efforts to go out, take a closer look and see if there's anything we can do to contain or eliminate the population. So starting with Brazilian waterweed, Aegeria densa and Hydrilla verticillata both look very much like our native Elodea. These are, again, not from here. They're native to South America, not found in Lake Champlain, not found in Vermont, but confirmed in surrounding states that are close by. So this Brazilian waterweed, I would say, the telltale is that it is a big plant and it has greater than 3 leaves on the whorl. And we'll get into that now. So our native Elodea canadensis only has 3 leaves on the whorl, 3 leaves consistently, and they are not serrated. So Brazilian waterweed, these leaves are not serrated, but there's more than 3. So if there's more than 3, um, this plant can get much bigger than our native, but we have to remember that we might be seeing the early growth and development of many of these plants, and they may look very different when they're just coming up than when they're fully grown. So the structure is important. You're looking for these lance-shaped leaves, and we're looking for greater than 3 leaves on a whorl, would indicate Brazilian waterweed. Okay, and Hydrilla verticillata is another very aggressive aquatic plant that looks also very much like our native Elodea canadensis, except it does have serrated leaves. And so here we're looking for greater than 3 leaves on the whorl, and they're serrated. So this invasive plant has a number of different forms. There's a monoecious and a dioecious form, and then a new northern hydrilla that is different from those in the Connecticut River. And we've intercepted all 3 of these different kinds of hydrilla coming into Lake Champlain from different water bodies on boats and trailers. So the threat is there. We hope it does not become established in the lake, but if you ever see anything, and you will see a lot of our native Elodea, you want to take a close look to make sure that the leaves are not serrated. Okay, so better close-ups. So again, lance-shaped leaf with these really clear tooth margins, and there's a rib along the backside of the leaf that also has spikes on it. And that's how we know this is hydrilla and not the Brazilian waterweed. So there's a number of different ways they can reproduce. They have turions, which are little buds that sit at the apex of where the leaves come out from the stem, but you may not see those turions until a certain time of the plant's development, usually a little bit later in the season. Some of the forms of hydrilla have these tubers, which is a really fleshy growth that is often buried in the sediment, and it's the overwintering mechanism for the plant. So some of these plants, if you were to remove them and get the roots with them, if you saw a tuber, our native Elodea does not have a tuber. Neither does Brazilian waterweed. So that would be an indication that it was a hydrilla plant. And then I think this one we have intercepted, and the leaf margins is really the toothed leaf margins is the telltale for this one. So that's what we look for, greater than 3 leaves. Our native lookalikes, and they look very much alike. Our common waterweed, Elodea canadensis, and then we have slender waterweed, which is Elodea nutali. And we can have crosses or hybridizations of some of these too, which can make it tricky. So um, again, you don't have to be the expert about Brazilian or hydrilla. All you need to know is our native has 3 on the whirl. If you're seeing something different, please let Eileen or myself know. And again, this is another really great side by side to remind us of the fact that we're looking for more than 3 leaves on the whirl for hydrilla and Brazilian elodea, and that for hydrilla, it's the leaves have that toothed edge. Brazilian does not, but again, 3 leaves on the whirl. And then the leaf vein has spines on hydrilla. Um, and so that's a really nice fold out that we have, and maybe could be provided to you too as a training tool.
It is in the Champ toolkits. You'll get your
Perfect.
own.
There you go. Okay, Carolina fanwort, Kabumba Carolina. Oh, we intercepted this year before last on boat coming up from Massachusetts. Um, this is another aggressive aquatic plant that does look like some of our natives too. It's not found in Lake Champlain yet. It is confirmed in other New England states. So we do have a lot to protect. I like to remind folks that we don't have a lot of these species yet. So this work is really critical. So yes, from South America, let's move on. I want to focus on how you can tell this plant apart from some of our native species. So Carolina fanwort, the way that we tell it apart is the leaves come opposite off the stem, but they also have that segment of the petiole or the leaf stem from from the stem until the leaf starts branching. Our other native species branch directly from the stem outwards and don't have that little leaf stem there. So it looks like almost like a fan, and these leaves do branch. So this is not a whorl around the stem. It's 2 leaves that come off and then start branching. Let's see. Oh, the flower's pretty. I haven't seen it in flower because we don't have it here. So um, I guess that would be another way that you could try and tell it apart. We do have some native submersed plants that look like Carolina fanwort. So our water buttercup, I've seen a lot of this in the northern end of the lake. Water marigold can be throughout the lake. But if you look at these leaves, they are attached directly to the stem and they branch from there. They don't have that segment or that stint before they start branching. So another good one to look at, and maybe underwater it could look like a milfoil, but when you get that leaf structure out, you'll see they're not feathers, they're actually branching and they come off the stem opposite each other. So if there is that section of that leaf stem before the leaves start branching, that's when the alarm bells should go off. So this one's a fun one to sort of put in your tray and take a closer look at. Okay, parrot feather, I've seen this one. We don't have it in Lake Champlain. It looks like a plastic aquarium plant. It is so rigid and it looks fake nearly. So these leaves are much more dainty. It's a rooted submerged plant. Yes, it's native to South America, not found in Lake Champlain, present in other surrounding states. One of the cool things and under this, zoomed in view is you can see that the flowers are right at where the leaves meet the stem and they just look like little cotton balls. They're really quite cool when you look at them up close with a hand lens or under a microscope. But the leaves are so rigid that this plant can extend up through the water and maintain its structure, which is unique. I've only seen it once in a pond, a private pond. Then we went in and took it out, and it is no longer in the state of Vermont. So I haven't seen it in other places, but it really does look like a plant. Uh, as the information shows, there are 10 leaflet pairs. So on every feather or so, there should be 10 little leaflets coming off of it. Round goby. Neogobius melanostromus is an invasive benthic fish that came over likely in ballast water into the Great Lakes. It was introduced in the 1990s. It spread throughout the Great Lakes, and it has been inching its way closer to Lake Champlain through the St. Lawrence Seaway and the Richelieu River to our north, as well as across the Erie Canal, coming into the Mohawk and the Hudson and trying to make its way up the Champlain Canal towards Lake Champlain. This fish looks very much like our native sculpin. And the telltale way to determine a goby from a native slimy sculpin is the pelvic fin. And the bottom of the fish, there's a fin. And if it looks like a suction cup and is all intact, all together or fused, that is a round goby. And it uses that suction cup to hold itself to rocks in some current. And the native or slimy sculpin has a splayed pelvic fin, so it's not fused. It doesn't look like a suction cup. There's really a lot of reasons why we're worried about round goby getting into the Lake Champlain. Primarily, it would likely decrease or eliminate our native biodiversity and outcompete the slimy sculpin, our tessellated darters, and other small benthic fish that other native species rely on for food. It really likes to aggressively eat eggs. We have a really great restoration story of lake trout in Lake Champlain, and this species in particular likes to hang out in those interstitial spaces in the rocks where lake trout would lay their eggs. Sturgeon and bass eggs would also be at risk. So they're likely to displace our native species. They're aggressive egg eaters. They can also carry viral hemorrhagic septicemia, which is a fish pathogen that can affect over 25 different species of freshwater fish. And we have not documented that pathogen in Lake Champlain yet. And they're really aggressive bait stealers. So when anglers have changed experiences on lakes where there are really dense populations of goby, such that when they cast their bait, whatever it is, they are continually coming up with gobies instead of the other target fish that they might be trying to angle for. So this one has the possibility of significant impacts to Lake Champlain, and we're working very hard to try and prevent that from happening. People will comment that round goby eat zebra mussels. It's true, they do. They would never eat enough zebra mussels to make an impact on the population that exists in Lake Champlain. Anglers will also argue that goby make bass and other sport fish bigger. This is true. If round goby reproduce and there's a large population, that is a source of food for some of our sport fish, and they could get larger. They may get larger at the risk of losing other native species and having other impacts to the fishery. So, for example, in the state of Ohio, the bass fishery, the early fishing season was closed because after round goby came in, the round goby would go eat all of the eggs on the nests while anglers were, in the time it took them to hook the bass, take the photo, put it back in the water, and swim back to its nest, all of the eggs were gone. And so they had to eliminate the early bass fishing in the state of Ohio for due to round goby impacts. So there are consequences, and it's often thinking and having more in-depth conversations about sort of holistically what these kinds of impacts could be to Lake Champlain. And then the quagga mussel is another species that is not present in Lake Champlain yet. It will not stand up on its side. If you have these, they have a lip on the underside that makes them sort of wibble wobbly. So you can see the zebra mussel on the left-hand side can stand on its side in a D shape. and the quagga mussel has sort of a ridge there. So if you're trying to stand it up, it would just fall over. The quagga mussels can get bigger than zebra mussels, but more importantly, they can reproduce and colonize in water much deeper than the zebra mussel. So we have a number of historic shipwrecks right now that are preserved in cold, deep water sites that the zebra mussels can't get to. And if quagga mussels were to get in, and there are mechanisms for that, we do have watercraft that come from the Great Lakes, whether they're loopers coming through the canals or being transported overland on trailers. Uh, this species could have some very significant impacts to Lake Champlain. We're surprised, but we don't have them yet, and we hope to keep it that way. They are in the St. Lawrence River. We do know that they can move through, the Erie Canal and get closer to Lake Champlain. And then starry stonewort. So Nidolopsis obtusa is a really fascinating plant that has this beautiful star-shaped barbell, which we will show you in the next few slides what it looks like. So this is a good size check here. But this isn't a plant, it's technically a macroalgae native to Eurasia. It was established in Vermont, and I remember when they first got it in like Memphremagog and going searching for it in Memphremagog. And it just looks like a very sort of low growing, it does look like an aquatic plant, but it's a macroalgae. And those little barbells, those little stars look like sort of hidden treasures in and amongst the growth. Um, but it's an important reproductive structure of the plant. So it's confirmed in New York and a lot of the Midwestern states. It's not as prevalent in New England yet. So this is definitely an early detection watch list species. So here's what the plant looks like. It sort of is, it's got less of a common structure in terms of it might be a little bit erratic in its growth. And you can see those internodal spaces between where the plant branches or the macroalgae branches varies. And there's different branching structures here. So this one, if you get this, it would probably look like a mat of macroalgae that you'd have to float and start to pick apart. Um, let's see. The whorls of 5 to 7 branchlets from each node. Now that is a lot. If you look at the node, you can see that there's varying lengths and varying dissections in the plant leaves that are coming off of the stem. Um, in order for this one to anchor to the sediment, it uses that a clear root, which is almost like translucent. And then at the nodes, there is that little barbell or a bulbil, that star-shaped. Uh, it's like a thick fibrous material. It's really fascinating. The science tells us we only have males in North America. I have only seen it in person once in Lake Memphremagog, so I don't have a lot of experience with it. But if you see this mass and those little bulbils, you're going to want to report that. All right, and water soldier. This one, um, is you can't miss it. Um, Water soldier is sort of it's a submerged species, but it has emergent leaves that are spiked. They will cut your fingers. This is a very dense, sharp, kind of nasty plant. Maybe from afar it looks like a common house plant, but it is not present in Vermont. We do know that we had a population in New York that was eradicated and in Quebec that is being managed. But this one would look like a spiky emergent plant coming out from underneath the water. Let's go to the next slide. So it has this sort of the lance shaped leaves are very long. They're serrated. These sharp spines and then white flowers. And again, I haven't seen them flowering before, but I think just the sharp spiked emergent leaves coming out of the water would be something that doesn't look like any of the natives we have in Lake Champlain.
Okay, so those are our 8 target and 8 watch list aquatic invasive species. Again, just want to reiterate, you don't need to internalize and have all of that memorized right now, um, or really ever, because we give you the full Champ toolkit that has a page dedicated to each of those species, and we give you tons of other identification resources that'll be useful for when you're out in the field. But those are our 16 species. I'm going to shift gears a little bit and talk about doing your survey, starting with the materials you'll need to do a survey. We'll provide you with a survey rake. It's this sort of double-sided, it's 2 rake heads attached to each other, which are then attached to a length of 25-foot rope. And I'll have some photos, and we'll be doing demos of that for our in-person training. Um, we'll want you to have a phone or another device with you, and that's for filling in your survey and taking photos. Um, we'll give you white containers, frisbees and or bins for this season. And that's again to have something to float your specimen in for photographing and for helping you identify the species. We'll give you bags for collecting samples and specimens, and we'll give you, like I said, identification resources. And we'll also give you a big tote bag that you can put everything in because it's kind of a lot of equipment. Um, and then if we do have 2 survey methods, you can survey from boat or from shore. Most volunteers survey from shore, but what's nice about surveying from a boat is it gets you out further into the water, so you're covering areas at a site that you wouldn't be able to from shore. But of course, there's the added hassle of being in a boat, so it's sort of up to you, and we'll discuss on what survey method works best for you, and I'll talk about both methods in this. Um, but if you do go out in a boat, you'll need a personal flotation device. So for those of you who have volunteered with Champ in the past, there's a bit of a change that we're making this season, and that's a switch to a different online reporting form. It's no longer through our website. It's on a platform called Survey 123. This should make things a lot easier because instead of filling in a paper sheet in the field and transcribing it at home, you can do everything at once in the field from your device. But if you prefer a paper data sheet and filling it in at home, that is still an option. I'll be bringing physical data sheets for the in-person trainings, and we can talk about what works best for you. Um, so for this training, I'm going to start with an overview of all the steps you take, front to back, for conducting a CHAMP survey. And then I'm going to switch over to the Survey 123 form so you can get a feel for what you'll be filling out in the field. So starting with the background section, um, you fill in your name and information. That's like your email, contact. You'll want to put the date of your survey and the time at which you are starting your survey. You fill in the name of your site and your survey start point. Now, CHAMP surveys take place between a start point and an end point. If you're surveying from shore, this is like a linear situation between these 2 points. And this tells us the area in which you covered. The start point should be the point at which you do your first rake toss. And a CHAMP survey consists typically of 10 rake tosses spread out throughout your survey area. Your site may very well not be a straight line along shore, even if you are surveying from shore, since you'll want to do rake tosses from if there's a dock there or if there's maybe some variability in the shape of the shoreline. Those are all just things that you can make a note of in the survey area description, which is like a text box in the survey form. And then you fill in some information about the water conditions, which I'll go over on the survey form as well. And then still standing at your survey start point, you filled in all that information, you'll do your first rake toss. Um, so for rake tosses, that's just throwing your survey rake into the water. It's a way to help you collect biomass. Biomass looks like this, what she is picking through in that photo. Um, but it's just a way to get whatever is in the water to you. And if you see aquatic invasive species in the water, but it's not coming up in your rake tosses, um, we still ask that you report it in your survey form. So you're not just reporting what you're pulling up, you're reporting everything that you're seeing during your survey. Um, and that would be something especially that would come up for zebra mussels. So if you're surveying like she is from a dock, you might not be pulling up much for survey for zebra mussels, but you might see, sections of the dock that are coated in zebra mussels. So you should still be reporting that. Um, but in order to do a rake toss, I'll show you how to do that in the field, but you just basically secure it on your non-dominant hand and throw it with your dominant hand. Then you pull it in at a slow to medium pace, collecting biomass along the bottom of the lake as you go. Um, so you did your first rake toss. Now you have this lump of biomass in front of you. That's when you sort of pick through it, and you examine individually what you're collecting from that rake toss. So um, at this point, you'll want to take your white container and get a little bit of water in there. And then as you're picking through that biomass, you can float new stems in there and figure out what species they are using your identification resources. Um, that helps with photographing them as well. So, as Meg had mentioned, this is a part that you'll want to be pretty thorough because you'll have the itty bitty zebra mussels attaching to plants, and you might also just have like kind of small fragments of plants that can come up too. Those can be tricky to identify, but that's where you can send us photos and collect samples. Um, so after your first rake toss, you'll want to do another 8 rake tosses spread out as evenly as possible over the survey area. And as you go, you're going to be filling in the aquatic invasive species that you find and characterizing their distribution. I'll go over the definitions of each of those terms um, when I go through the survey 1, 2, 3 form. And you'll also, as I mentioned, want to be doing a visual scan of the water in addition to what you're getting on your rake. As you're picking out aquatic invasive species, you can go ahead and not toss them back into the water. You can let them desiccate on shore. Um, and for native species that you're picking out, you can put them back in the water. Um, and it's very important here that you photograph at least one example of each aquatic invasive that you report. So if you see zebra mussels, you don't need to take a picture of every single zebra mussel that you see, because that is probably not possible, but you can take a picture of one of them that you're pulling up in your rake tosses, or you can take a picture of the patch that you're seeing from shore. And that goes for all the species. So just one representative example photo of each of the species that you report. Several photos are welcome, in fact, encouraged, but one is required at least for per species. Um, and using that white container is the best way to take photos just because it helps us see the little differences in the plant structure and help identify it. And what's cool is I'll show you how to do this, but on survey 1, 2, 3, you can take photos directly through there. So if you're filling out your survey in the field on a device, you don't have to switch out of the browser. You can do it directly on survey 1, 2, 3. So you've done now 9 rake tosses. Your 10th should be at your end point. And that's you do your last rake toss and you mark where that endpoint is, fill in the final bits of your survey, adding in any notes that you need to, and then recording that point in the end time. And then take a gander at your survey, make sure if everything's filled out, and then you can click submit. And this photo is another good example of how to float a specimen in a bit of water to help with identification. That's Eurasian water milfoil. Just a couple notes on if you do choose to survey from a boat. Um, there's a lot of equipment associated with doing a CHAMP survey. And having all that out with you in a boat and with paddling, it can be a lot. So if you do survey from a boat, I suggest going with a buddy. Some volunteers, what they do is collect their rake tosses as they go and set it aside, and then they'll do a full species inventory on shore with everything combined. If you do choose to do this, make sure that you know as you go the distribution of the aquatic invasive species. So whether they are trace, sparse, moderate, or dense. Um, and another note on surveying from a boat is rather than going on a straight line along a shoreline or, following the contours of the shoreline if there's a dock, what you want to do is follow this serpentine path within a survey area. So kind of snaking around, and the end of that survey area, as far as like how far out you'll go in the water, should be about to where the littoral zone of the water ends. And the littoral zone is the space where plants grow in the water. So it's where light is reaching through the water column. That varies depending on your site. There's some sites that can go shallow for a really long time. So, just use your best judgment. You don't need to go out a mile into the water. Um, but following about basically where plants are growing near the shore. And then what you'll want to do for marking that on your survey is making a note of those start and end points. That's where your snake takes place within. And you can estimate about how far out you go in the survey area notes. Something else I want to flag is that we did this last season too, but every volunteer we're asking to collect and submit a quality assurance sample, aka a QA sample. This allows us to make sure that everything that you're calling an aquatic invasive species in your survey is actually that and not a native lookalike, and vice versa, that you're not missing any aquatic invasive species in your survey. I suggest doing this for the first survey, not only because it gets it out of the way, but it's also a really good tool for you for identifying things that are common at your site, because I'll share with you what I identify from the QA sample. Um, and that's for both native and invasive species. You'll want your QA sample to be representative of your site. So that means that you want to give examples of each distinct aquatic invasive species that you're encountering during your survey, as well as the native species that you're finding in the survey. So you may need to pick things out of different rake tosses as you go. So if you find your Asian water milfoil on just one of the rake tosses, you'll still want to put it in that QA sample. Um, and but yeah, you'll see there's steps there on how to label it and how to bag it. Um, you'll want to get like a little wet rag or something in there to help it stay wet. And if you forget that, you can just do a little bit of water in the bag. Um, and then seal the bag and keep it cool. And uh, we'll exchange the sample with either LCC personnel or with a boat launch steward. I'll go more into detail about that. Um, okay, so Survey 123 is a software for data collection. The CHAMP survey form is basically just a multiple choice form that you can do while you're out there, and you can upload photos and mark your exact survey start and end points directly in there. Rather than having to switch around and figure out your GPS situation, you can do it right in the app, which is nice. And when I say app, I mean you can use your phone's browser. So you don't need an account to do a CHAMP survey, and you don't need the Survey 123 app to do a survey. You just need um, either your phone or an iPad. And again, we have options for folks that prefer not to do this method and like the transcribing at home. And we can talk about that during the in-person trainings. The link to the Survey 123 form is going to be on our email communications with you. It's also going to be on this volunteer landing page that we're putting on our website, which has everything that you need as a CHAMP volunteer. And it's also going to be a scannable QR code in the CHAMP toolkit. So when you have your Champ toolkit out there with you, you'll be able to get to the survey form from your phone just by scanning it. Um, so, and I want to reiterate, this is our first year trying this platform for Champ. So if you run into issues, please contact me and we'll help you submit your survey data. Um, just want to test it out, I guess. So I'm gonna just go through that survey form. Let's make sure that this works. I'm actually gonna. So you'll first confront this screen, and unless you are somebody who uses Survey 123 a lot and you have the app on your phone, I prefer to just kind of click open in browser for this part. Um, can everybody see that? I know you're all muted with mic off, but can
Can
everybody see the survey?
can you make it bigger?
Yes, I'll zoom in. Is that a little better?
Yes.
Okay, cool. Thank you. I started filling this in just for the training, but this would all be blank when you pull it up. So uh, from the jump, fields marked with a red asterisk are required, so you won't be able to send in the survey if one of these are not filled in. Um, so you want to put in your first and last name. If you were with somebody else, you'll want to also put their name. Um, put your email address, and most of your affiliations will be LCC volunteer, so thanks. You want to put the date of your survey. Oh, the mouse is going crazy. Um, and you can do that. It has this calendar drop down. You just select the date and the start time. You can do that also with this drop down, or you can just type it in. Um, you'll type in your site name and the town or city of your survey site. Um, there's a few different ways to record your survey start point. You can start by using your current location. So when you click this, it should hopefully load up to where you are currently. So this zoomed in on the LCC office because that's where I am right now. But you can zoom out over here. And for this survey, I'm saying that I did Blanchard Beach at Oak Ledge, which is this section of shoreline. So I want to be very precise. So I toggle over to where my site is. And also, if you don't want to share your location, you can enter an address here. You can click current location or you can type it in. You can also just sort of move the map around to get there, or you can enter your latitude and longitude. Um, and yeah, you want to zoom in and be really precise on where that start point is. So I am saying I started here. All you do is click at that start point. And also, if you're filling this out in the field and you share your location, you it will snap automatically to where you are. So you don't even necessarily need to move it around. Um, but once I get my start point in there, I then go to the survey area description. So you just want to have like a couple sentences sort of laying out where your survey area takes place within. You can be as descriptive as you'd like. It's also very helpful to talk about things that are like permanent structures or landmarks just to help orient the person who's interpreting the survey. Because say you find something like a watch list species, it's then helpful to know precisely where you were so there can be follow-up. Um, then you mark your survey method, either rake tossed from shore or from a boat. Relative water level and clarity. These are estimates. So it should be relative to your site for the time of year because water level will fluctuate with the time of year. Um, so you can just give your best estimate for both of those, um, particularly if you don't know what the usual is for that time of year. Um, And then for light conditions, you're describing the weather, and surface conditions, you're describing the texture of the water. So those of you who are cyanobacteria monitors are familiar with that parameter. So if the water is still, you put calm. If there's a bit of movement, you put rolling. And if there's enough rolling that you're seeing white caps in the water, so like little crests of waves further out, you put white caps. So that's all the background information. Then moving on to the target aquatic invasive species, it asks if you've just seen any of these. These are those 8 species that we know are in Lake Champlain to some extent. When you click no, you can move on. When you click yes, you get a drop down that consists of an inventory table. Sorry. So yes. Uh, and for this survey, let's say I find like a couple curly leaf pondweed and one Eurasian water milfoil over at Blanchard Beach at Oakledge. There's definitions for these distributions categories right here. So if it's not present, you put not present. If it's a couple of species, you put trace. If it's somewhere between that and fewer than 10, or thinking about zebra mussels, like a single small patch of zebra mussels, you put sparse. For moderate, that's if you have over 10 individuals or a medium size or a few small patches of zebra mussels. And then dense is so many that you're having trouble counting the number of individuals. Or if you have a very large patch of something like zebra mussels. So you fill in this whole table, and it's important that for the things that you're not seeing, you put not present, because with the red asterisk, we see that that's all required. And then everything that is marked as something other than not present, you'll get this automatic pop-up for photos. So basically anything that you report, there's going to be a photo section. You need to attach at least one photo of each recorded species in the corresponding section. So you can do this from photos that you've already downloaded. Please ignore all these other photos I have. Oh, that's curly leaf pondweed and Eurasian water milfoil. You don't have to go in and name your photos. I know that that's something that cyanobacteria monitors do. It's a nice thing to do if you feel like it and have the time, but no need to do it. And if you do choose to do it, you can do it right in there by going to rename. And you can also edit what you put in by hitting delete there. And you can use this additional photos field to attach photos of the surveying process. If you go out with a buddy and you get a picture of them surveying, that's always fun to get. Get photos of the site that add context or a view of the aquatic invasive species population from shore, really anything that gives good visual context to your survey. It's not required, but it's welcome. And another thing to note is that if you want to put multiple photos of the same species, you have it'll keep coming up with new drop-in options. So I'm doing this from my desktop and I'm attaching photos that I already have downloaded. But when you're in the field, you can attach photos also by going to this button here, and it allows you to take a photo right then and there, which I recommend for doing your field surveys. And then scrolling down further, you'll come to your watch list AIS section, and it asks if you've encountered any of those 8 watch list species. It's the same thing. So when you select yes, you'll get this inventory table that you'll want to fill out with all of the distribution metrics for each species. The only difference between this section and the target species section is that if you do report a watch list species, we also ask, and there's a reminder about this directly in Survey 123, we do ask that you collect a sample of what you're reporting. You can do that with one of the Ziploc bags that we provide you. You label the bag with your name, the date, your site, and fill in this info. So say you encounter Brazilian waterweed, I think it should pop up here. So you'll put in that photo, and then in the notes section, you can put some details about what you found, and then your availability for passing on the sample to either us or to the Lake Champlain Basin program. That's just because we want to make sure that we're doing the early detection rapid response follow-up if you are finding watch list species. So this makes it a little bit easier to do that. So then we get to the section on quality assurance samples. Again, the steps to do that are nice and here, so you can be reminded. And you mark if you collected one, or if you've already done one, you'd put that, or if you're going to do it on a different survey, you put that. But if you select yes, there's another pop-up section that will talk about how you plan to submit your QA sample. And there's three main options for that. You can either drop it off at our office on Flynn Avenue in Burlington. We're in the office pretty regularly, Monday through Friday, 8:30 to 4:30, but there is some variability, particularly with the summer field season. So it would be nice to get in touch to schedule a drop-off time just to make sure someone's going to be there to get it. And you can do that. My contact information's all right there. And then if for those of you who are kind of further from Burlington, like Champlain's pretty big, so Burlington's kind of a hike for some of our volunteers. You can also drop them off with a Lake Champlain Basin program boat launch steward. We have specified boat launch sites and times and days that are good to drop things off. And I will share that with you as soon as I have it. It will either, and I'll put it on the survey form and also on like the volunteer landing page and in all my communications with everybody. So you'll have like a good set schedule of where you can go to drop it off with a boat launch steward. If neither of those work, we can coordinate another alternative to have me come pick it up, either if I'm already traveling near you, or we can sort of find like a midpoint to do a pickup. So that's the QA sample section, we'll say. And then getting to the end of the survey, you'll want to fill in notes on your survey. It's not required, but just sort of describing the things that you found. It's nice to have this section to sort of give an explanation that isn't necessarily captured in those distribution metrics. And if you talk to people about Champ, that's always a fun thing to mention in your survey notes. And then same idea for the survey endpoint. So it'll go straight to my location again, I believe, and then I'll scroll over to where scroll over to, we'll say that I ended my survey at the end of this speech. So there. And it's the same thing, you just click. And the survey end time, this will snap actually automatically to the time that it is. So make sure that you check, I'm saying that I finished this at 1 o'clock. Pay attention to that AM PM distinction. I did not finish surveying at 1 a.m. So I've filled out everything and I just take a look, make sure everything's looking hunky-dory and accurate and I didn't miss a field. What's nice about Survey 123 is if you did miss a field, it will not let you submit. So once I think everything looks good, I click submit, it'll upload. And it's the same thing for when you're doing this from your phone or from your tablet in the field. I'll give that a second because you'll get like a little, yeah. So once you send it in, you should get this message and that's how you know that it went through. If you don't get it, let me know and we'll work through it. But that is the Survey 123 field. And again, if you have any issues, be it with like that submit button or with anything else throughout the survey, please contact me. Okay. Okay, so we're only 7 minutes over right now. I'll try to wrap things up quickly with the next steps. If you the next steps now are we'll email you a welcome package which will have this volunteer landing page, which has the champ manual and everything you need to submit data and all the additional identification resources. You'll be getting physical copies of all that too for the in-person training. And the in-person trainings are all listed there. You should have signed up for one for when you signed up as a champ volunteer. But let me know if you need to change that. And for those of you who selected that none of these trainings work for you, I will be in touch to schedule additional trainings. And then at those in-person trainings, we'll discuss choosing survey sites. So for those of you that are new or maybe want to change your survey site, we'll figure out where works best for you then, and we can begin surveying after those in-person, after a volunteer finishes the in-person and the virtual training, you're good to go. Okay. That's a lot. Are there any questions? You can either ask them in the chat or you can just unmute yourself and fire off. Let me open the chat. That might be helpful.
Will we get access to the recording of this session?
Yes, yeah. So part of that like welcome email that I will aim to send out tomorrow, but definitely this week, will be a link to a full recording of this training. And any other questions? Yeah, I'm hoping Survey 123 goes well this year. It's a really great platform and automatically maps all of the data, which is really cool. And I'm excited to share like the visuals that we're able to get from that at the end of the season. Okay.
Thank you all. We look forward to seeing you out there this field season.
Yeah. If more questions come to you that aren't coming to you right now, you can contact myself here or Lindsay here. And all of our contact information will be in our outreach to you. And with that, yeah, thank you everybody so much for attending this training and for working with us this season. We have a really great volunteer response this season, so I'm really excited to see what y'all find and work with everybody.